All right, hi everyone. My name's Eden. As Dr. Mann mentioned, I'm an undergrad senior at Duke University, and this past summer I was part of the summer research training program at the Myon Lab at Mount Sinai. So today I'll be presenting on identifying therapeutic targets from RNA sequencing of senescent cells using a few different tools that have been developed by the lab. So I'll start just by providing some background on cellular senescence and why we're interested in it. Then I'll introduce the Appyter tool that was used to identify senescence targets, and I'll demonstrate how we selected a set of potential protein targets for senescent cell removal. Finally, I'll just highlight one of these targets, and I'll present some exploratory enrichment analyses that connect the top genes that we found. So cellular senescence is a state of permanent cell cycle arrest, and it's been implicated as a key contributor to aging. There's recently been a large body of evidence suggesting that removing senescent cells through senolytic therapy, it might be an effective treatment against a lot of different age-related diseases. So as far as we know, aging-related senescence it's induced by a pretty complex variety of stressors. And there's definitely variability by cell and tissue types, but there is evidence for common molecular markers across these heterogeneous senescence phenotypes. So in this project, that's kind of what we were interested in looking at. The specific form of senescence that we chose to study is called replicative senescence, or RS, and it recurs as a result of most of our cells' limited capacity to divide. So this is known as the Hayflick limit. And during aging, we believe that there's just more and more cells reaching this limit and entering a state of senescence. So one of the reasons we chose to focus on RS as opposed to other forms of senescence has to do with an interesting difference between mouse models and humans. So mice, which have been like a popular model used to study senescence in vivo, they've been shown that RS specifically occurs by different mechanisms in marine cells compared to human cells. So in humans, it's pretty much directly caused by telomere shortening, but in mice, for a variety of reasons, including their much longer telomeres, RS seems to be induced by oxidative stress and some other environmental factors. So by studying RS in human cells, we hoped to just capture some senescence targets that maybe aren't as well known from mouse models of senescence. Similar to cancer, senescent cells become pathological when they start just being able to evade clearance by the immune system. So in normal senescence processes, they should be routinely cleared. And so as a result, there's been a lot of interest in senolytic immunotherapy to treat age-related conditions. But a big challenge for just immunotherapy in general is being able to identify cell surface proteins that can actually facilitate targeted cell removal. So for instance, using ADCs or CAR T cells. And in 2017, Boss et al., this was a group, they published an interesting study where they developed a prioritization algorithm that compared RNA-seq data from a patient's tumor to normal RNA-seq from the GTEx consortium. And their goal was to identify targets that were only highly expressed in the tumor with low expression across all other normal tissues. They also prioritized cell surface proteins, which in general are more ideal candidates for immunotherapy. So using this approach, they were able to identify a candidate protein, which they targeted with an ADC. And this allowed them to safely and effectively deliver cytotoxic drugs that eliminated neuroblastoma cells both in vitro and in vivo. There have been a few similar strategies that have been used to identify markers in senescent cells. And these two research groups also demonstrated successful targeting with ADCs and CAR T cells of those senescence targets. So inspired by the concept described by Boss et al. And a few of those other research groups, the Myon Lab, we wanted to develop a publicly accessible tool that could facilitate this type of identification and visualization of targets with some added depth and also some more user options. So this tool is called the Tumor Gene Target Screener Appyter. It was mainly developed by Daniel Clark, who's a data scientist in the lab. And how it works is a user can upload transcriptomic data from a sample to identify targets for any disease or biological condition of interest. So for this analysis, we applied the Tumor Gene Target Screener to find targets in RS cells specifically. My own contribution to this Appyter was adding visualizations of protein expression from 3 different protein atlas efforts. So having protein expression from normal cells and tissues is really useful because we know that mRNA expression of gene candidates doesn't necessarily correlate with protein expression. So with proteomics data, we can have some additional evidence for the safety and efficacy of a gene's coded protein as a drug target. Proteomics data from non-disease tissues were retrieved from 3 different proteomics atlases. The first, the Human Protein Atlas, the Human Proteome Map, and also a GTEx proteome project. So now in the Appyter, the user can view the protein expression levels from different normal tissues and cell types to identify if there's any areas in the body where a candidate protein has low expression and it might be a safer target. So as an overview, the aim of this study was to identify targets, senescence targets using the Appyter. Our workflow was to first process some senescent samples against a few different backgrounds of normal human cells and tissues at both the gene and transcript levels. And then we validated these targets by looking for an association with aging using data from the GTEx consortium. So first, to gather senescent cell data, we searched for studies providing RNA-seq from multiple replicates of RS cells. And in total, we analyzed 13 different data sets from 6 in vitro studies of senescence. And something just to note is that by design of many of these in vitro senescence studies, most samples happen to be from human fibroblasts. The transcriptomic data from each of these studies was processed by the Appyter for targets, and then we aggregated the results from the 13 data sets. So this is kind of what the Appyter interface looks like. First, we selected to analyze the senescent samples at both the gene and transcript levels. These samples were processed against 3 normal tissue backgrounds, which were bulk RNA-seq from GTEx and ArchS4, and then single cell data from Tabula Sapiens. So this single cell data set was added to the Appyter by another fellow summer scholar, Reid Fleischman. We also decided to prioritize membrane proteins because of their immunotherapeutic potential. And we wanted to check that the protein expression levels of the candidates are low in normal tissues. So we selected this option to visualize protein expression with those 3 proteomic resources. And then with these parameters, the Appyter performs differential expression analysis using Lima. It returns the candidates that are highly expressed in the senescent sample, along with these requested visualizations. So each senescent cell sample was analyzed for the top 20 targets. And initially, this produced just over 600 membrane protein coding genes that are highly expressed in at least one senescence analysis condition. We also filtered this list by checking for agreement at the transcript level. So essentially, gene targets were kept if one of their transcripts were among the top 200 transcript targets in any analysis condition. In the second step, it also allowed us to identify if there's any splice variants or isoforms that might be specific senescence markers. We then also filtered for targets that were consistent across multiple conditions. So we removed any proteins that weren't common targets across different RS samples and normal tissue backgrounds. And then finally, because we're interested in cellular senescence, specifically in the context of human aging, We wanted to identify targets whose expression is elevated in older compared to younger tissues. So to do this, we used the tumor gene target screener to identify transcripts highly expressed in older tissue samples from GTEx, so ages 70 to 79, relative to tissues ages 20 to 29. So we took the top 200 transcripts from these 47 different aged tissues and cell types as like kind of our reference for finding age-associated targets. There were 29 final membrane proteins that met these criteria. So these were top differentially expressed at both the gene and transcript levels, followed by consistency across multiple analysis conditions, and then finally association with aging. So these are the top differentially expressed membrane proteins that we found in RS, and the genes highlighted in red are those that are age-associated. We see that all three normal tissue backgrounds, and so ArchIS4, GTEx, and Tabula Sapiens, they're all represented among these analysis conditions that identified the top targets. These are the targets that appeared in the most conditions, along with their differentially expressed transcripts and the tissue in which the transcript showed a significant association with age. Each candidate was also searched for direct links to cellular senescence in literature. And so while most of the top consistent targets are supported by literature as highly expressed proteins in senescence, there's a few of them that we found to be novel candidates. So as an example, a target I'd like to highlight is programmed cell death ligand 2, or PDL2. It binds to an immune checkpoint receptor, which is believed to activate T cell exhaustion. And in various studies of cellular senescence in different tumor types, PDL2 expression, it was found to be significantly upregulated both at the mRNA and the protein level. And upregulation of PDL2 is known to produce immune evasion in senescent tumor cells. There's also a study of mouse models that found PDL2 to be overexpressed in aged mice compared to younger mice. So in this analysis, at both the gene and transcript levels, we found that PDL2 is highly expressed in RS cells compared to all three normal tissue backgrounds. We also found that the protein coding transcript of PDL2 is highly expressed in older fibroblasts and adipose tissue compared to all younger tissues. There was a research group that found that inhibiting PDL2 allowed senescent tumor cells to be cleared by the immune system. So now PDL2 is actually patented by this group as a drug target for removal of damaged and/or senescent cells. Antibodies for PDL2 are also produced and available commercially, so there's potential there for ADC development. And then lastly, as you can kind of see from the proteomics data shown here, protein expression of PDL2 is actually elevated in many normal tissues, with the exception of connective and soft tissues. So although it probably wouldn't be a safe universal target for removal of senescent cells, it could still be considered as a candidate for targeted removal of senescent cells in adipose tissue or some other soft tissues. An app that I want to highlight is the Enrichr Knowledge Graph, which is a prototype bioinformatics app that was initially developed by Nhi Nguyen, who was another summer research trainee, and is now being hardened by Errol Evangelista, who's a bioinformatician in the Mayan lab. So Enrichr-KG, it can be used to see how genes might relate to each other, as well as to certain phenotypes or diseases. And this is especially useful for exploring any novel targets. So for instance, shown here is ITGA11, which was one of the novel drug target candidates identified for senescent cell removal. And from the knowledge graph, we can see that there might be some relation with the PI3K-AKT signaling pathway, which we know is a well-known driver of cellular senescence. As an exploratory analysis, we were also interested in identifying if there are any pathways or phenotypes that are highly represented among the top genes expressed in RS cells. So we submitted the 32 most common targets across analysis conditions to Enrichr, and this was without membrane prioritization. And Enrichr identified a few significant phenotypic associations among these common senescence genes, including terms that are related to reduced bone mineral density and abnormal immune response. So these are phenotypes that are consistent with the potential role of these targets collectively in some aging-related processes. An interesting association we also noted is that this gene set is enriched for genes downregulated with the JAG1 molecule. And there's evidence that JAG1 drives cellular senescence or drives senescent cell clearance in aged human skin through this Notch1 signaling pathway. So the senescent cell samples that we analyzed, there might be some suppression of pathways linked to JAG1, which is in line with the ability of senescent cells to evade immune clearance in these age-related pathologies. So in this study, we identified both novel and known candidate targets for senolytic therapy using the tumor gene target screener Appyter. One limitation to note about our approach, though, is that differential expression analysis, it doesn't guarantee that the protein actually has low expression across all normal tissues. But proteomics can be used to further evaluate the efficacy and safety of the target. Overall, we just demonstrated how the tumor gene target screener Appyter can in general be used for accessible and informative computational screening to identify immunotherapeutic candidates from transcriptomics. And just as a side note, and I suppose as suggested by the name of the Appyter, the tumor gene target screener was mainly developed for application in cancer. So my summer internship, I did apply a similar approach to identifying targets for the CPTAC3 pan-cancer cohort. And the nice thing about this approach with the Appyter that wasn't really demonstrated in this project is that we can use it to identify any patient-specific targets for one patient or cluster-specific targets if we're interested in a subgroup of patients. So as some next steps, the targets we identified here would, of course, need to be validated in some follow-up experiments in vitro, including development of ADCs. Also, these RS targets should definitely be investigated in other forms of senescence, as well as senescence in different cell types and different stages of senescence. And finally, since these targets would eventually also need demonstration in animal models, we want to extend this screening approach to the mouse genome from where it can be more directly tested in vivo. So Daniel Clark actually recently added the Tabula Sapiens data set as a new background in the Appyter, and we definitely plan to be adding more soon. So I want to thank my fellow summer interns, Reed Fleischman and Yin Nguyen, as well as Daniel Clark for all of their amazing contributions to this project, and a big thanks to Dr. Mayan for his great mentorship. And I think at the end, I'll gladly take any questions, and thank you all very much for listening.